Okay, good morning. Today we're going to continue our discussion of PCR reactions, going over a few things that are a little bit hard to understand, and then move on to talk about DNA synthesis, DNA sequencing, the Sanger method. Okay, so PCR and sequencing are both examples of in vitro DNA synthesis, which is very different from what happens in a cell, although it still behaves according to the regular rules of DNA replication. You need to move 5' to 3' on the new molecule, which is to say you're always adding on to 3' hydroxyls on the sugar. You need to have a template. You need primer. You need deoxyribonucleotide triphosphates, as well as the DNA polymerase. Okay, so PCR reactions are a lot different also from what happens in a cell, because we're going to do repeated rounds of synthesis on a specific template and therefore get a lot of a particular DNA sequence. We're not just making one copy and moving on. We're going to reiterate the process and get a gazillion, sometimes literally a billion, copies of the sequence that you started with. Okay, now what's unusual about PCR and also about sequencing, it's selective in the sense that you are choosing the synthesis that you will be doing by virtue of your choice of primers. In a cell, we need to replicate all of our DNA, so the process is very different. Here, we can select out whatever small parts of the whole DNA we have to be synthesized based on the fact that that's the only place that our primers are going to bind. And again, it involves cycling among different temperatures to allow the reactions to occur. Okay, so here's the picture we had last time, and it's a very schematic version. It kind of lies a little bit because in reality, when you do a PCR reaction, you mix everything together in one microfuge tube and let the machine do all the work of the heating and cooling and stuff. The primers are already in there, the polymerase is already in there, your deoxyribonucleotide triphosphates are in there, and the machine will go ahead and heat and cool and time the various steps of the reaction. Okay, but the essence of what's happening is well illustrated. You have a region of double-stranded DNA that you want to amplify. You heat to separate the strands, that's usually about 94 degrees centigrade. Then you cool, and the primers that are already floating around there in the reaction mix can bind to their complements on your DNA strands, your template. Okay, and so you need two types of primers, one for each strand, because it's only going to hybridize to that one section of that one strand and not the other. Okay, the other thing that you have to realize is that these primers are present in huge molar excess, so there's plenty of primers to go around for any templates that need to be amplified. So after you anneal the primers, you have to heat it up a little bit because the heat-stable polymerases that are used don't work well at the 55-degree hybridization temperature. They work best at 72 or 68, depending on what particular polymerase you're using. So when you get up to the warmer temperature, you will then have DNA synthesis extending across the template, beginning at the primers, and so you make two new strands, one for each of the template molecules. Now each of the new molecules starts with a primer and is included in it, so your PCR product will have a primer on the 5' end of each strand. Okay, so you denature at 94, roughly, cool usually to around 55, and then you warm it to allow this DNA polymerase to work, and then you repeat the cycle again, denature, cool, warm, etc., usually about 30 times. That's sort of represented here, and you can see kind of what's happening in the reaction. You have your double-stranded DNA, you melt it, the primers will anneal, you extend the primers to create new strands. Then the whole process is repeated. Denature the strands, allow the primers to anneal, and those primers are then extended to create two more new strands. And again, this is the third round, heat, anneal, extend to get more of the same products. So this is an amplification kind of situation rather than just a synthesis, because each time you create a new molecule, it then becomes a template for the synthesis of yet another new molecule. So the amount of new DNA that you're producing increases exponentially. Here you've got 2 new strands, 4, 8, etc., going on towards 2 to the 30th, ideally. Now, I don't think you really do have 2 to the 30th new molecules after that many rounds of synthesis, but you do have many, many millions of copies, at least, of the DNA that you're trying to replicate. Okay, this is a different representation of the same thing, but it illustrates one part of the process which sometimes confuses people. And so I want to just look at actually what's happening to these templates. Okay, we have our template DNA, which is actually much, much longer than is shown. It could be an entire huge piece of chromosomal DNA. But it gets melted, the primers will anneal, and then beginning at the primer, it extends. The new molecule is extended. Both of the strands will become extended. Okay, now I call these stragglies because even though they have a fixed beginning position defined by the beginning of the primer, one for each of the two strands, there's no fixed end position. There's nothing to make it stop. It just kind of runs off until the polymerase finally decides to fall off the template. So that's not a defined product yet. We have to wait to the next cycle to get the defined product, which begins and ends with primer sequences. We need another cycle to get that to happen. Okay, so each time we have a reaction, we'll make another straggly, but when the straggly itself serves as a template, that's when we get the defined product. So in this second round, you melt, extend, and create a new strand for in each case. So this is where the defined product that begins and ends with a primer starts to form. Now those will later take over the reaction, because once that molecule is made, you've lost what's before and what's after it in the original template. It starts and ends with either a primer or a sequence that's complementary to the primer. So when this little guy itself replicates, it doesn't have the parts that stick off the ends. It can only be replicated in and of itself. So that's the defined product. It starts with a primer at the 5' end of each of the two strands. But when those start serving as templates, that's all you can produce is more of those. And those guys take over the reaction, because you can only make 30 rounds if you're a template. You're going to get one straggly, and that straggly is not going to, it's only going to get made once. It's never going to make it again, because if it's made again, it's going to turn into a defined product. Now, the way to convince yourself of this is to sit down with a pencil and paper and the primers and just draw it out for yourself to convince yourself that that's true. So in the end, the two strands are complementary to each other, fully base paired, and the primers are at the 5' ends of each of the strands, and they are part of the products. Okay. Which of the following temperatures in the PCR reaction does primer annealing occur best? 94 degrees, 72 degrees, 55 degrees. Yes, 55 degrees. Okay, some characteristics of primers, right? They will select for us the region we want to amplify, which presumably is something we want to look at in more detail, because their binding to the template will provide the start point for DNA replication and select the region of interest for us. Okay, so they have to bind the DNA in a complementary way, and they have to point their 3' ends across the region of interest that we're trying to amplify. So you need two primers, one for each strand. These are called the forward and reverse primers. The forward one is the one that binds to the left part of the sequence as written, and the reverse binds the right. So it's important to choose the sequences and polarities to get the desired product. Okay, so here's our hemoglobin example again for convenience. Okay, again, we're only showing one strand, and we want to choose primers that can just amplify this particular DNA sequence. Okay, so the forward is going to bind to the left, and the 3' end is going to point to the right, and the reverse for the reverse. Okay, so in order to figure out what sequences and polarities we need to remember that it's double-stranded structure. So I'm going to write that and then show what the primers should be for this region. Okay, so all I did was manually take that top sequence and complement it to make the opposite strand. So I can illustrate that or make it a schematic by drawing the two strands, 5' to 3', with the understanding that those are the sequences of those two strands. So now we need to design our primers, assuming we're going to start at the very end of each of those two molecules. Okay, so this illustrates what you should be thinking about. You need to have your 3' hydroxyl pointing across the region that's going to be synthesized. So the way that will work, of course, this is a 3' to 5' strand on the bottom. You need something to bind to it, complementary to it, with its 3' hydroxyl available for DNA polymerase to then extend across that region, making new DNA. So that's our left primer, or our forward primer. For our reverse primer, the same thing, only with opposite polarity. We need to have our hydroxyl pointing towards the left so that that primer can then prime synthesis of a complementary strand to that upper strand. So taking that and relating it to the actual sequence that we have, we can realize what those sequences are going to be, right? So we need, for example, for the lower strand, we know that we're going to have write something that's complementary to it in order to prime synthesis across it. And also for the upper strand, we need something complementary to it with the correct polarity to drive sequencing across from it. So what that looks like really is that the forward primer is the same as the upper sequence, and the reverse primer is the same as the lower sequence, because both are complementary to this, similar to their same complements. So it's the job of the forward primer to create new upper strands, because when it goes across that region, it's going to make another upper strand. And then similarly, we need the reverse primer to create more lower strands, because when we synthesize across the region, that's what we'll be creating. So you need one primer for each strand, correct polarity. Okay, so it's not all as simple as all that, because not all primer pairs work very well. And this is not something you need to worry about for this class, but in case you're worried about this in real life, you certainly don't want the two to base pair with each other. That would be bad. Then you make a big primer rather than an actual PCR product. They should not have a lot of secondary structure. If they have a lot of internal base pairing, they might prefer to do that rather than to hybridize to the template, which is what you want it to hybridize to. Another consideration is you want the two to have similar melting temperatures. If one is 55 and the other is 70, they probably won't work very well together. Also, they should be unique. If you're amplifying from genomic DNA, you want to make it unique to that particular sequence, not something that binds all over the genome. That would make kind of a mess. So you really want to have sequences that are as unique as possible in that genome, if that's what you're amplifying. Okay, so to make life easier for you, because this can be a little intimidating, there are places where you can analyze this for free, fortunately. They're the same ones that will sell them, so they want you to analyze it and then buy it right away. But these are very helpful. If you don't, calculating melting temperature in particular is quite simple for these guys, but not as easy if we try to do it ourselves. Okay, so usually making a PCR reaction is just the first step towards something more complicated, because all you have at the end of a reaction is a test tube full of DNA, you hope. Okay, so it has to be analyzed or used in some way following this. Now, sometimes it's just as simple as running it on a gel and looking for its size, but more often it involves sequencing or cloning or other sorts of things. So we'll have some examples next week of where we would start with a PCR reaction in order to learn something else, but that can wait till next week. The other thing we'll talk about at some point later in the class is something called qPCR, which is also known as real-time PCR. So in regular PCR, you stuff your sample in the machine and go away for 2 hours or whatever while the amplification happens, and then you take it out of the machine and then run it on a gel or something just to make sure that you've got what you want. So this is an endpoint, really. You don't know what the kinetics were of accumulation of the products. You just know that the product is correct and it's the right size. So if you want it to be quantitative about it, you need to follow the reaction as it's actually occurring. And there are instruments that will enable you to do this by looking at fluorescence as it increases over the course of the reaction. So that's very important for using to detect RNA, actually. RNA can be used as a template, but not directly. First, it's copied to make a single-strand cDNA, which is then, in turn, used in the qPCR reaction. But in any case, we will deal with this on some later day. Okay, so the next topic of interest is in vitro synthesis for Sanger sequencing, and the requirements are very much the same as for PCR in the sense you need a template, you need a primer, and you need to have, of course, nucleotides, right? The slight difference here is that, again, you need to know something about the sequence that you're going to be sequencing in order to actually carry out a sequencing reaction. And that seems a little bit circular, but you need something to get the reaction to start. Now, if it's a PCR product, it's pretty simple because you know what primers you use to create the product in the first place. So you can simply use one of those as a starting point, starting primer for your synthesis reaction. Or else it might be something that's cloned into a vector where we know what the sequences are on the ends. In any case, for your primer for DNA sequencing, you need to have a fixed start point whose location and identities are known. All right, so different types of templates are used. Could be a piece of human DNA cloned into a plasmid, could be a PCR product, etc. It differs from PCR in the sense that you're only going to synthesize one strand. You're not going to try to do both at once just because of the way the reaction works. So even though it involves DNA polymerases and DNTPs, you're only going to do one strand at a time. So you need DNTPs, you need a DNA polymerase. But what's unique and crucial for this sequencing type of reaction versus PCR itself are these chain-terminating nucleotides. Now, these are very fundamental to the mechanism whereby we will determine our sequence. So we're going to use these to read our sequences, basically. So what these are dideoxyribonucleotide triphosphates. We also will need a method for examining the products after they've been made, and that's going to be capillary electrophoresis. And this is capable of resolving molecules that differ from one another by only a single nucleotide, which is going to be very important in a minute. Okay, here's one picture of the chain-terminating dideoxys. Okay, and we're showing three types of NTPs here. On the left, we have the ribonucleoside triphosphates, which is what you see in RNA, where your 2' and 3' carbons are both linked to hydroxyl groups. And then we've got regular DNA, DNTP, where we have the 2 carbon, it's got a hydrogen on it, which is why it's deoxy. But of critical in DNA synthesis is the 3' hydroxyl, which is what will be used to add on new material in the process of a DNA synthesis reaction. So that OH is critical to be able to extend a primer across a template, because without that, no extension can happen. Okay, so key here is our dideoxyribonucleotide triphosphates, where that critical OH is replaced simply by a hydrogen. And we will be using these for our reactions, as well as normal DNTPs. Okay, these are also labeled with fluorescent compounds. You have to use 4, of course, one for each nucleotide type, and each one is labeled with a fluorescent compound such they actually emit light at different colors. So you can distinguish one from the other based on the fluorescence, based on the color of, in this case, an elongated DNA molecule. Here's another picture of your 2', 3' dideoxy. You can see both hydrogens here. So once this is incorporated during synthesis, no further additions are able to happen on that molecule. All right. I think I said all this. Okay. Okay, so as always, we will have a template shown on the bottom of the slide, and we will have a primer that specifically binds to that template such that it can then be used to extend across the template to determine the sequence. Okay, so this type of reaction is going to involve a mixture of regular nucleotides plus a small amount of the dideoxynucleotides. Now here, the purpley looking one is dideoxy ATP. In reality, all 4 of the dideoxys would be present in the same mixture. Okay, so what's important is you've got a mixture of the normal ones and the dideoxys, all 4 of them, all in one reaction, such that at any point along the synthesis, the polymerase has a chance of incorporating a dideoxy at that position, thereby terminating that particular chain. Now realize that this is happening to about a gazillion molecules all at once, so stopping one molecule doesn't stop the overall reaction, it just stops the synthesis on that particular molecule. So really there would be all four, so you'd have four colors in here, and that would allow polymerase to choose at each position whether it's going to put in a dideoxy or a regular nucleotide. Importantly, when you get the dideoxy incorporated, there's no further addition possible on that molecule. So in the polymerase picture shown, that molecule is done because it ends in a dideoxy, and so that is going to define one product of the sequencing reaction, which starts at the 5' end of that primer, extends to the dideoxy A, which is that reddy purpley color, and we will be able to see that in our capillary electrophoresis. So I want to illustrate this a different way, including all four, to show you what happens as we work our way across a template. So this is pretty much what I just said, I believe. Right. Each one is fluorescently labeled with a different color. That color tells us that that nucleotide is an A, and that's going to be important later. Okay, so here's a goofy drawing. Okay, I put my template in the opposite direction, but you get the idea. Here's a template. We want to synthesize DNA across it in such a way that we can determine the sequence. So as part of it, we have, of course, normal nucleotides of each kind, but we have a mixture of the 4 dideoxynucleotide triphosphates, and each one is labeled with a different color. So at the first step in this reaction, we're going to have this primer here bound to the template with its hydroxyl exposed to polymerase. It's, of course, going to put in a T opposite that A, which is the first nucleotide in the template. So at this point, the polymerase has a choice. It will either incorporate a regular T, in which case the reaction can proceed past that point, or else it will include a dideoxy T, at which point it will stop. So we will generate at least some molecules that start from the 5' end of the primer and end at that very first nucleotide in the template. So here's this, and here's what happens. Okay, we can possibly put in a red dideoxy T or a black unlabeled regular one. So this will be our first sequencing product. It has a length beginning at the 5' end of the primer and going to that T. Right, so we can imagine a small molecule that we've produced. This illustrates that it's going to go very fast on our gel. It's the smallest one. The next position is going to be a C, and so we can either incorporate a dideoxy G or a regular G. If we get the dideoxy, the reaction stops, and we create a band or a product which is one nucleotide longer than the first one. So we can put that one step up on our imaginary gel here with a blue G that will be one nucleotide difference in length from the first T. But it can also get a regular one, in which case it will continue to the next nucleotide, which in this case is going to be a G, right? So we can put in a C dideoxy, in which case we'll get a green band, or else we'll put in a regular nucleotide, in which case the reaction will continue past that point. But that third product is a C, and so we end up with a band, a green band, which is one nucleotide longer than the previous one. So we're going to make a step ladder, our imaginary gel, with bands in increments of one nucleotide as the polymerase marches its way across this template. So the next one after this is going to be a T, etc. Right? So what we're going to do is make this ladder, essentially, of bands, incrementing by one nucleotide each. And by analyzing the colors of each, starting at the bottom, the 5' end, working our way one nucleotide at a time to our 3' end, we will determine the sequence of its molecule. So in terms of analyzing this, this is drawn as if it were a gel. It could be a gel, but mostly it's a capillary now. But the idea is the same. You denature your samples, so you're only looking at the one strand. And then you put them on the gel and allow them to migrate through the gel. But instead of stopping the electrophoresis at a particular point and staining it, what you do is you put the molecules on there, let them migrate, and they can migrate all the way through the gel pass this laser beam and detector. So what's going to happen is when the molecules make their way through the gel, they will at some point encounter the laser, which will excite the fluorochrome in the fluorescent molecules, and that fluorescence emission will be recorded and then identified by the very complicated software that says what it is. So we can imagine these are marching along, and marching along past the detector, past the laser beam, each one separated from the other, so that you'll get a pattern of bands or pattern of colors corresponding to what nucleotide it is. Okay, now these are usually reported as a trace, which is a little bit non-intuitive, but if you imagine the molecule as it approaches the laser, there's going to be a position where there's no fluorescence, and all of a sudden the band will start to be influenced by the laser. Then you'll get an increase in fluorescence corresponding to the middle part of the band, and then a decrease in fluorescence as the band moves along past the detector. So what this is what is recorded as is a series of peaks, which are assigned different colors according to the identity of that fluorescence molecule. Okay, so here's one representation of it. This is represented as bands, where you can read the sequence from the colors as they march past the detector. All right, so more typically, this slide's correct, but it's kind of too busy. This is easier. All right, so more typically, what you see is a series of peaks of different colors, where each color tells you what the nucleotide was at that position. So this is reading 5' to 3', with the shortest molecule being first, and then the getting longer and longer to the point of several hundred nucleotides usually. Now the machine knows what color is. You don't just sit there reading colors. It will tell you what the actual nucleotide is corresponding to each one of those peaks. So a sequencing trace is typically what one would get, along with a text file saying the actual just called bases. Here's another sequencing trace. And remember, it's going 5' to 3' along the synthesis, and that's what's being reported by the machine. So it doesn't seem exactly fair because we're synthesizing the molecule and we're reporting the sequence of the thing that we're synthesizing. We're actually reading across the template, but we're not recording, we're not saying that's the sequence of the template. This is the sequence of the newly synthesized molecule because it depends on the colors that were incorporated by polymerase as a reaction proceeded. So it doesn't seem fair, but that's what's the way it's done. You're always adding on to 3' ends. So if you read across the trace, you're starting at the 5' end of the new molecule and extending towards the 3' end. The term dideoxy refers to sugars that lack the OH groups on which of the following carbons: 1 and 2, 2 and 3, 1 and 3, 3 and 5, 2 and 4. 2 and 3, yes, that is correct. Dideoxy NTPs are used for PCR only, DNA sequencing only, both, or neither. Answer: DNA sequencing only. Both strands of DNA are synthesized during PCR, DNA sequencing, both, or neither. Answer: PCR. 2 primers are used in the reaction. PCR, DNA sequencing, both, neither. Answer: PCR. Primers are made of RNA. PCR, DNA sequencing, both, neither. Neither. Very good. Okay. Have a good weekend.